Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
As cells perform their everyday functions, they turn on a variety of genes and cellular pathways. MIT engineers have now coaxed cells to inscribe the history of these events in a long protein chain that can be imaged using a light microscope. This technique could help shed light on the steps that underlie processes such as memory formation, response to drug treatment, and gene expression. There are a lot of changes that happen at organ or body scale, over hours to weeks, which cannot be tracked over time, says Edward Boyden, the Eva Tan Professor in Neurotechnology, a professor of biological engineering and brain and cognitive sciences at MIT, a Howard Hughes Medical Institute. If the technique could be extended to work over longer time periods, it could also be used to study processes such as aging and disease progression, the researchers say. One way to study these functions is to image proteins, RNA, or other molecules inside the cells, which provide hints to what the cells are doing. However, most methods for doing this offer only a glimpse of a single moment in time, or don't work well with very large populations of cells. Biological systems are often composed of a large number of different types of cells. For example, the human brain has 86 billion cells, Lingu says. To understand those kinds of biological systems, we need to observe physiological events over time in these large cell populations. To achieve that, the research team came up with the idea of recording cellular events as a series of protein subunits that are continuously added to a chain. To create their chains, the researchers used engineered protein subunits, not normally found in living cells, that can self-assemble into long filaments. The researchers designed a genetically encoded system in which one of these subunits is continuously produced inside cells, while the other is generated only when a specific event occurs. Each of these tags can bind to a different fluorescent antibody, making it easy to visualize the tags later on and determine the sequence of the protein subunits. For this study, the researchers made production of the V5 containing subunit contingent on the activation of a gene called c-Fos, which is involved in encoding new memories. We're hoping to use this kind of protein self-assembly to record activity in every single cell, Lingu says. It's not only a snapshot in time, but also records past history, just like how tree rings can permanently store information over time as the wood grows. Recording events in this study, the researchers first used their system to record activation of c-Fos in neurons growing in a lab dish. To explore whether this approach could work in the brains of animals, the researchers programmed brain cells of mice to generate protein chains that would reveal when the animals were exposed to a particular drug. Later, the researchers were able to detect that exposure by preserving the tissue and analyzing it with a light microscope. The researchers designed their system to be modular, so that different epitope tags can be swapped in, or different types of cellular events can be detected, including, in principle, cell division or activation of enzymes called protein kinases, which help control many cellular pathways. The researchers also hope to extend the recording period that they can achieve. In this study, they recorded events for several days before imaging the tissue. The total amount of information it could store is fixed, but we could in principle slow down or increase the speed of the growth of the chain, Lingu says. If we want to record for a longer time, we could slow down the synthesis so that it will reach the size of the cell within, let's say 2 weeks. In that way we could record longer, but with less time resolution. The researchers are also working on engineering the system so that it can record multiple types of events in the same chain, by increasing the number of different subunits that can be incorporated. The research was funded by the Hock E. Tan and K. Lisa Yang Center for Autism Research, John Doerr, the National Institutes of Health, the National Science Foundation, the U.S. Army Research Office, and the Howard Hughes Medical Institute. Recording of cellular physiological histories along optically readable self-assembling protein chains. Nature Biotechnology, 2023, doi, 1038, S41587, 022, 01586, 7 Massachusetts Institute of Technology. https://ift. tt. 36 EMA1W. Access January 2, 2023. Thanks for watching. Please subscribe my channel for more videos.